Welcome to Is Pharmacology Difficult podcast. I'm your host, Dr. Radhika Vijay, MBBS MD Pharmacology, and this is the audio hub to get the best simplified basic tips, strategies, methods, and lots of ideas to learn better, understand better, and make your concepts crystal clear. If you really find and if there's a question hovering in your minds, is pharmacology difficult? Lend me your ears for a while and let in the magic of knowledge. So today we're going to start off a new season. Yes, I know it's been a long break and we are going to begin with a new season, a very nice, knowledgeable, informative one. And any guesses? What's the topic? Well, let's not waste time. The topic is all about screening, evaluation, drug discovery, and a lot of such related topics. So, today we're going to start off with the introduction to all these topics, and I'm going to cover a few aspects of the drug discovery. That's the introductory part. And later we're going to go for individual screening of the drugs. That's going to be a brief aspect of each of the screening of each drugs and drug groups. But yes, later on in some of the coming times, I may give you a detailed description also. But in this season, we're going to cover everything in a little concise manner and comprehensive manner that won't make you run away from the screening of the drugs. So without any delay, let's get to it. So first of all, let's understand the drug discovery. Well, drug discovery is a very important process. Now, there is a necessity of the fields like medicine, biotechnology, chemistry, and pharmacology quite intertwined together. Initially, most of the drugs, they were discovered by serendipity. Most of you know that. If you have been a student of pharmacology, you know what is serendipity and you know how the drugs were discovered. And there was a constant effort and process of identifying the active ingredient, the active constituent in the traditional plants and the medicinal plants. We know that so many discoveries were there, so many efforts were there. According to the classical pharmacology, substance or anything having a desirable therapeutic effect is to be identified in the form of a medicine or drug. And later on, in the process of the reverse pharmacology, what is going on? The sequencing of the human genomes, which were actually in the process of very fast cloning. And then they led to the synthesis of ample amount of purified proteins that were identified. So that was the process of reverse pharmacology. Then there's something known as capital HTS, what we identify as high throughput screening. It is a very useful, very important method. It allows us to learn about the screening of a large number of compounds. So especially when the targets are isolated compounds, then such methods are very, very useful and they undergo the process of disease modification. Now let's come to the modern drug discovery. What is that concept? It identifies a lot of optimization techniques to hit the screen. Screening, hitting, optimization techniques. And how they are useful. These techniques finally lead to enhanced affinity, bioavailability, and also enhanced and very good drug half-life. And also it is accompanied with the decrease in the adverse effects. Then we know now we have computer-aided learning and drug designing also. So this is also a very important process. It is actually a revolutionary process in the drug discovery. Now once we understand all the complications and the procedures and the physiology of the biological systems and the various accompanied advances and developments in the technology, then we are very well able to understand the whole drug discovery process in the present era. But we should understand this whole process is very, very long process. It's very costly and actually a kind of not very efficient one, but still It's quite a hectic process. Now let's come to the process. What is the actual process and what are the different steps and how does it go about? Now, there is a concept of rational drug design. That is, we need to identify and find out new medications or drugs based on the knowledge of the biological target. That is the proper or the apt or the rational drug design. Now, generally what happens in drug designing, a lot of small molecules are involved. They are complementary in shape and charge to the biomolecule target, and they interact due to this structural similarities, charge similarities. They interact with the target and finally they bind to it. They have affinity for it. They bind to it. Okay? What is a drug? What do you understand by the term drug? It's a small organic molecule which can activate, stimulate, enhance, or inhibit, decrease, or stop the function of a target biomolecule. And generally these target biomolecules, these are proteins. And that finally leads, whether activation or inhibition, it finally, what is the outcome? What is the benefit? The therapeutic effect. That is the whole benefit of this whole interaction. Interaction of the drug with the target biomolecule protein finally leads to therapeutic effect. Now, let's understand a little process about the time that is taken in the drug discovery. It starts with the target discovery. That takes around 2.5 years. Then the second step is the lead generation and optimization. This whole thing takes about 3 years. Then there's a process of preclinical development that takes about 1 year. Then there are phases of different clinical trials like phase 1, phase 2, phase 3 clinical trials that takes about 6 years. That's a long process, and because of the length, the cost also enhances. Then the next step is the FDA review and the approval. That takes 1 year. And finally the drug reaches the market, and then follow-up is done, post-marketing process start, and that is total 14 years. Now you can understand how hectic, how lengthy, and how costly it is. Now I'm going to talk about the steps in the drug discovery. That is important. Now I'm going to talk about the steps in the drug discovery. The first step is the target identification. Now in these steps, what is the combination? The combination is both of knowledge of and around the receptor and the random screening process that allows the drug discovery. You should have a thorough knowledge of these things, and then you can understand and go and proceed further in drug discovery. So target identification is the first and the foremost thing. You should first and foremost identify a suitable drug target. What can be the target? The target can be a receptor, an enzyme, a nucleic acid. Now, for an example, morphine reacts with a molecular target in the body, and that leads to the detection of the new targets which the morphine interacts with. Okay, so that is how we identify the interaction of the drug with the target biomolecule and identify it. Then, with the knowledge of genomics and proteomics, one could identify in the earlier situation a lot of drug targets. And still, the research is going on to identify more and more drug targets. The more selective the drug, the less it will interact with the other target drugs, molecules. And the higher selectivity of the drug will lead to less adverse effects of the drug. I hope you understand this thing. Now, if you're a student of pharmacology, you will understand this language and terminology very, very easily. See, for a very good example, a very selective drug is penicillin. It targets a specific target in the cell wall. It targets a specific receptor or target in the bacterial cell wall. And that's why it is not targeting anything else. So it's very selective drug. Now, you should know about the target specificity and the selectivity within the body. If a particular drug is inhibiting an enzyme, it should inhibit the target enzyme only, not the other enzymes. Okay? That is the specificity and the selectivity of the target. Then we know about receptor agonists and antagonists. They should be selective for a particular receptor. Like, you know about adrenergic receptors, you know about a particular receptor subtype in adrenergic receptor, like beta 2 adrenergic receptor. Very selective. Okay? So selective drug will act on beta 2 adrenergic receptor. Now targets can be specific organs and tissues in the body. Like in the heart, in the cardiac region of the our body, we have beta 1 adrenergic receptors, while in the lungs we have beta 2 adrenergic receptors. Sometimes a particular drug can have multiple targets. One of the very good examples is olanzapine. It targets many receptors: dopamine receptors, serotonin receptors, adrenergic receptors, histamine receptors, muscarinic receptors, and it is not a selective drug. It's a very non-specific, non-selective drug. So, drugs which interact with a large range of targets, like olanzapine, they are called promiscuous ligands or dirty drugs. So, this is a new term. If it's a new term, note it down in your notebooks and keep it in your mind if you learn something new. Now, to end up today's session, I will be talking about the structural properties of drug molecules. They can be physical properties or physicochemical properties. First of all, let's enlist the physical properties. They can be shape, stereochemical properties, electronic properties, physicochemical properties. And specifically, if we talk about physicochemical properties, they are the role and the structure of the water on the drug structure, solubility of the drug molecules, drug partition coefficient, and the surface activity of the drug molecules. These are the physicochemical properties of a drug molecule. The clinical other properties that are considerable while looking for a profile of a drug are bioavailability, drug hydration, etc. So these were a list of the different properties of the drug which are very, very involved in deciding and judging about the specificity and the selectivity of the drug. So we have to be very careful. We should know a little bit about the properties. We should know that why a drug is behaving in such a particular manner, why it is targeting a particular target, why it is targeting multiple targets, and then we have learned some new terms today. So all these should be kept in mind. Till the next time, for all the updates and latest episodes of my podcast, do visit www.pharmacologydifficult.com, where you can subscribe to my e-newsletter, Pharmacology for the e-newsletter, and you can also follow for more updates. If you're listening for the first time, don't forget to subscribe and follow whatever platform you are consuming this episode. Stay tuned. Do rate and review on iTunes, Apple Podcasts. Stay tuned. Thank you. Take care. Bye-bye.